Tēnā koutou ko Vivian Hunter ho, no mai, haere mai, ki te Aotearoa New Zealand Food Safety Science and Research Centre, ka nui, te mihi nui, kia koutou katoa. My name is Vivian Hunt, and on behalf of the New Zealand Food Safety Science and Research Centre, I would like to welcome you all to our third Spotlight on Food Safety Science seminar. Today, I'm delighted to welcome Dr. Kang Wang from the School of Chemical Science at the University of Auckland. Kang, who lectures in food science at the university, is a research scientist who uses an interdisciplinary approach to food safety applied to food processing. He draws on mathematical modeling, material science, microbiology, and molecular imaging with a focus on sustainable practice in food systems and aquaculture. Kang has a PhD in biosystems engineering from Zhejiang University, and after graduating there in 2013, he joined the University of Massachusetts Amherst for his postdoctoral research. He worked on biomaterials and biointerfaces. In 2015, he joined the University of California, Davis to explore new technologies to improve food safety. He worked at UC Davis for 5 years on research at the interface of food engineering and food packaging. And at the end of 2019, he made the wise decision to join the University of Auckland. So we're very pleased to have attracted Kang to share his knowledge and experience with New Zealand food scientists. So Kang will speak today about his research that looks at sustainable bio-based interventions eliminating cross-contamination of perishable foods. But before he starts, can I just remind you to have join in with your audio rather than video, so we're not sure about Zoom and its capabilities. And if you have any questions, please send them to me via chat. Just select my name and type in your question, and we will answer these at the end of the seminar. So Kang, over to you.
Thank you, Vivian. Thank you for your introduction. Good morning, everybody. My name is Kang, and it's my great pleasure to be here and have this opportunity sharing some of my previous research experience with all of you. So I think if you are interested in my work, I would be more than happy to discuss more, and also more than happy to collaborate with any of you. So today I'm going to talk about part of my previous research projects, which is related to the food safety. And before we start today's topic, let's just look at some recent news. So we know the foodborne outbreak is a critical issue over the world, and you can see in 2018, there's the E. coli outbreak linked to Roman lettuce happened in the United States and Canada, and which resulted in almost 200 infections, and also a lot of people going to hospital. And also last year, last Thanksgiving, there 140 vegetable products were recorded due to the listeria in contaminations. And also in New Zealand, last year in May, the sprouts product we have an outbreak linked to the salmonella contaminations. So the microbial food safety is very important, and it's related to everyone, and especially as more and more consumers like to eat fresh or minimally processed food. So we need to keep all the food without the passenger contaminations. So the WHO estimates 1 in 10 people got foodborne infections every year, and which resulted in a lot of people going to hospital and more than 400,000 deaths. And also it causes significant economic loss, which is higher than $55 billion every year. So among all the foodborne outbreaks and foodborne illnesses, the number of foodborne outbreaks linked to fresh produce is higher than any other single food categories. So why the fresh produce are very easy to be contaminated. So in our study, we have tested the different scenarios of cross-contamination of the fresh produce. So I know there are a lot of literature reported the cross-contamination study, but usually the contact time and the contact force are not precisely controlled. So we have designed a device which can precisely control the contact force and contact time, which enable us to get more accurate results. So first scenario, we are looking at the cross-contamination from a contaminated leaf to the clean leaf. So the results tell us the cross-contamination can happen instantaneously. And after 1 second contact, we can get more than 3 log of bacteria transferred from the contaminated leaf to the clean leaf. And the second scenario, we are looking at the cross-contamination from the leaf surface to the food contact surface. In this case, we test the glass surface, the stainless steel surface, the plastic surface, which are the commonly used food contact materials. And after 5 seconds contact, we can see all the material can get more than 3 log of bacteria transfer from the contaminated leaf surface. And also the hydrophobic surface are more preferred by the bacteria to attach. So the third scenario, we are looking at the cross-contamination from the wash water to the leaf surface. So in this case, we are looking at 2 situations. The first situation is there is no organic particles in the wash water. So the wash water was contaminated by the free bacteria. And the second situation, we are looking at the role of the organic particles in the wash water on the fresh produce cross-contamination. Because we know that usually in the produce industry, there are a lot of organic particles in the wash water, such as the plant debris. So we are looking at the role of those particles. So again, we can see that the results told us that without the organic particles, we still can get more than 3 log of bacteria transferred from wash water to the fresh produce surface after 1 minute exposure. But if we have the organic particles in the wash water and those particles were inoculated by the bacteria, then we can get more than 4 log of bacteria transfer from the wash water to the fresh produce. So that means the presence of the particles enhances the bacteria transfer from the wash water to the fresh produce surface. So based on those 3 scenarios, we know that the bacteria, the fresh produce, the bacteria attachment happens very fast, instantaneously. We can get more than 3 log of bacteria transfer. So how can we remove the bacteria from the produce surface? So when I worked in California, there is a lot of larger fresh produce industries in the Salinas, and we visited a lot of industry companies. And for example, the Taylor Farms, they use the open flume washing system. Hansing, they use turbulent flow system, and the Tarati, they use the pipe type washing system. So they have different designs to enhance the bacteria removal from the fresh produce surface. But unfortunately, all of them can only get less than 2 log reduction from the produce surface. So the reason is we know that the produce industry, they usually just use the chlorine-based sanitizer because chlorine is very cheap, and they just add more and more chlorine to their wash water, and then maintain the free chlorine level around 15 ppm or 20 ppm. But the for the fresh produce, there is high organic load on the produced surface, so which will consume most of the sanitizers. And also the complex surface roughness and the surface chemistry will protect the bacteria from contact with the sanitizer. And also if the biofilm forms on the fresh produce surface, which will make the bacteria more resistant to the sanitizers. So based on those introductions, we know that the cross-contamination happens very fast. And once the cross-contamination happens, the bacteria on the produce are very difficult to remove. So we know that the major source of the bacterial contamination of the fresh produce, it could be from the contaminated produce, it could be from the contaminated wash water, and it could be from the contaminated food contact surface. So in my study, we developed the low-cost food-grade antimicrobial delivery system to enhance the decontamination of fresh produce. And we developed a natural food-grade compounds-based sanitation approach for the rapid sanitation of wash water. And also we developed the hybrid antimicrobial and anti-foreign materials to reduce the contamination of the food contact surface. So let's just look at the first part, the smart sanitizer, the antimicrobial delivery system. So this work was quite interesting, and we filed a patent application for this work, and also we received the USDA grant for this work. And we got some exciting results. So in this case, we are using some cell-based particles, and we encapsulate the antimicrobial polymers into those particles. And then if we charge those particles, then we can get the chlorine compounds bind to the polymers. So we can get the antimicrobial particles. So I know there is a lot of studies reported regarding the antimicrobial nanoparticles or microparticles, but the unique feature of our formulation is that we are not modify the particle surface. We are load the antimicrobial compounds into the particles, so we can get a localized high concentration of the antimicrobial compounds. And on the other side, our formulation has a high affinity to bind the bacteria, so we can achieve a targeted delivery of the antimicrobial compounds. So based on those two key benefits, localized high concentration of antimicrobials and also the high affinity to bind diverse microbes, the hypothesis is if our particles attach to the leaf surface, then it will find the bacteria and then attach to the bacteria cells. And then some of the free chlorine will be released, but most of the cells, bacteria cells, will be killed by the direct contact. And also due to the enhanced shear stress caused by the particles, we can not only kill the bacteria on the fresh produce surface or on the food contact surface, we can also remove those dead bacteria from those products. So the first key benefit, localize the high concentration of sanitizer. So we can load 5,000 ppm of chlorine on our particles. And also if we store our formulation at 4 degrees C for 1 month, the chlorine content didn't decrease significantly. And also, we know that the organic content is a big challenge for the chlorine-based treatment. In our case, we at the organic load in the wash water at the level of 2000 COD level, which is a typical organic load in the produced industry wash water, we didn't see any significant reduction of the chlorine content. But if we expose our formulation to the extremely high organic load, which is the 20,000 COD level, then we still can get 80% of the chlorine remain on the particles. So the second key benefit, our formulation has high affinity to bind diverse microbes. So in this case, we tested the binding efficiency of our particles to E. coli, gram-negative bacteria, gram-positive bacteria, Listeria, and also the fungal cells, the Candida. The binding efficiency of our formulation to all the microbes are higher than 20%. So which means the binding efficiency is pretty high. And some applications, if we use this formulation to treat the biofilm on the food contact surface, in this case, we grow 4 days E. coli biofilm on the plastic surface. Then we can see after 20 minutes treatment, we can get 7 log of bacteria reduction. But the conventional sanitizer, chlorine-based sanitizer, we can only get 2 log reduction. And also, as we said before, our formulation not only kills the bacteria, we can also remove the bad bacteria from the surface. So another application, use our formulation for the treatment of fresh produce. So in this case, we are comparing with the chlorine-based sanitizer, which is 50 ppm, and also we use our formulation with the equivalent chlorine content. And we found that the chlorine-based sanitizer for the treatment 5 minutes washing, we can only get less than 2 log reduction, which is consistent with the industry data. But if we use our formulation, we can get 3 log reduction. And if we extend the treatment in time, then we can get all the bacteria killed by our formulation. I know someone will argue that those high concentration of chlorine will cause some quality degradation on the fresh produce surface. So we tested the chlorine residue on the chlorate and the perchlorate generation during the washing procedure. And we found that if we use the 50 ppm chlorine, the conventional chlorine-based sanitizer, we can get significant increase of the chlorate compounds. But our formulation, the results are very similar to the unwashed fresh produce, the lettuce leaf surface. So which is pretty good. And also we tested the texture, color, and the other quality properties. We didn't find any significant changes. So another application regarding the reduce the food spoilage. So we know that the botrytis is a problem for the tomatoes or for grapes. So we used our formulation to treat the botrytis mold. And we found that the negative control after 5 days, we have the we can find the mold on the produced surface. And if we inoculate the botrytis first, then spray the free chlorine, 100 ppm free chlorine on the tomato surface, we can get mold. But if we use our formulation, even 50 ppm chlorine content, we can achieve a very good results. We didn't get any mold after 5 days incubation. So there, based on those 2 key benefits, we can have other applications. For example, we can immobilize our particles on the food contact surface by either by chemical conjugation or by the physical entrapment, create a coating on the food contact surface. And also we can use our particles to as filling materials for the filtration systems. And also we can create edible antimicrobial coatings on the cleaning, like the polyfoam sponges or the brushes, which is typically used in the fresh produce industry. And also, I want to mention that our formulation is a very low cost formulation because, for example, if we use this yeast cell based formula particles, then we can get the spent materials from the winery industry. So we are using the food waste, basically speaking. So the cost will be very low. And the second part, I would like to introduce a rapid sanitation approach for the wastewater. So in this case, we are using the low concentration of natural antimicrobial compounds and also combined with the low level of physical processes. So in this case, the hypothesis is if we use a very low concentration of natural food grade compounds, we didn't get any bacterial reduction. This low concentration means the ppm level of the concentration. And if we use the sublethal level of processing technologies, like the UVA light or the mild heat, the temperature lower than 55 degrees C, we didn't get any bacterial reduction. But if we combine them together, then we can get a synergistic antimicrobial effect, which can help us to achieve a significant log reduction. And in our lab, we have discovered a bunch of this kind of compounds, and they have different categories. But in this case, I just take one example due to the interest of time. So the key benefit of this approach is it can replace the abrasive chemical agents by the natural antimicrobials and will help us to create a clean label and also reduce the sanitizer concentration. So we can achieve a rapid microbial inactivation while lowering the processing impact on the food quality. So I just take curcumin as one example, which is a widely used food grade compounds in the food systems. So in this case, if we use 5 microgram per milliliter of curcumin and combine with the UVA light, so if we don't apply the light, then we didn't kill any bacteria. But if we turn on the UVA light, then we can achieve a significant log reduction. And also, it's not necessary to use UVA light. We can also use LED blue light, the cool white light, and even the sunlight. We can get the same results. And also, this approach can be working at both room temperature and the refrigerated temperature. I know the fresh produce industry, they usually operating for at 4 degrees C, so which is suitable for the fresh produce industry. And also, this approach can be working at different pH values, and also especially for the lower pH level, it will be working very well. So this property will be very good for the juice treatment because usually the fruit juice, they have the lower pH values. So again, we are testing the inactivation of bacteria in high organic content in the wastewater. And we find that if we have the 1000 COD level, then we still can get more than 5 log reduction in 10 minutes treatment for both E. coli and Listeria. And also another application for this work, we know that in the grocery store, the fresh produce shelf, they usually have the spray systems. So if we use this kind of spray system to deposit the low concentration of curcumin, in this case, we use 10 ppm then for 10 minutes. And then we turn on the light. It's not necessary to use the UVA light. We can also use some normal light, LED light, or even the sunlight for 5 minutes. Then we can get 2 to 4 log reduction on the spinach surface, the leafy green surface, and also on the tomato surface. So besides the sanitation of fresh produce, the processing of the juice, this work can also be used for the irrigation water treatment, because on the farms, we already have the sunlight. So if we just add this kind of low concentration of compounds, then we can sanitize our irrigation water to prevent the cross-contamination. And also this can be worked for the solid system, the solid water disinfection for the drinking water. So the third part, I would like to introduce some antimicrobial materials that we have been working on. So the first part is the hybrid antimicrobial and antimicrobial coatings on the stainless steel surface. This work, we are collaborating with the dairy industry, and we developed some this kind of modified stainless steel surface for their processing equipment, because we know that the accumulation of foreign materials on the heat exchanger plate is usually a big issue for the dairy industry, because it will increase the risk of cross-contamination and also reduce the heat transfer efficiency. So in this case, we incorporate 2 kinds of nanoparticles on the coating, the nickel phosphorus coatings on the stainless steel surface. And we can inhibit the biofilm formation, and also we can kill the bacteria attached on the stainless steel surface. So from this image, we can see the native stainless steel surface, so we can get the biofilm formed on the surface. But the modified surface, we didn't get any biofilm formed on the surface. And this work was highlighted by the Food Safety News, and also this can be used for the packing tables for meat or for the apples, and also for use for the medical supply to modify their surface. So another work is the antimicrobial and anti-foreign plastic surface, plastics. This work, we were collaborating with the Center for Produce Safety, and they want to develop some liner materials for the fresh produce, because we know that the fresh produce bins or totes, they are used multiple times every day. And if some fresh produce are contaminated, so the bacteria will be quickly transferred from the leaf surface to the bin surface or tote surface. And also, if we once we get a new batch of the produce, then the bacteria will be transferred for the new fresh produce surface. So we can design some liner material put between those 2 items, then prevent the cross-contamination. So in this case, we use the again, we use some chemical approach to bind the chlorine compounds on the plastic surface. So let's look at this figure. If we have the our material, then if bacteria attach to the surface, then some of the bacteria, most of bacteria will be killed, and they will consume some of the chlorine compounds on the plastic surface. But if we expose our used liner material into 1% bleach, then the chlorine the film will be recharged by the sanitizer. So they can be used for multiple times. It's a rechargeable material. And if we use this material for as a packaging material for the strawberries, it can prevent the food spoilage. So we can see here, this group, the first group is without the plastic membranes. And we found that after 2 weeks, all the strawberries got mold. And if we, this control group is the strawberry were covered with our plastic membranes, but those membranes don't have the chlorine content. So we can see still most of the strawberries will get spoiled. But if we use the chlorine charged membranes, then we can find only one strawberry got mold. So it can be used for extending the shelf life of the strawberry. And also, as we mentioned before, we immobilize our yeast-based particles on the plastic surface. Then this material can also be rechargeable. And we tested multiple times, quenching and charging, then the chlorine content didn't decrease. And also, we are using this material for the test of biofilm formation. We can see on this native plastic surface, we can get significant biofilm formed on the surface. But for this one, these cells are just yeast-based cells. So there's no bacterial cells on the plastic surface. So in the next 2 slides, I think I would like also to introduce some of my other works. This work is related to the aquaculture industry. So we are collaborating with BASF, a very large chemical company from Germany. So we are developing some edible phage-based coatings on the fish feed, because usually we know the fish industry, they use the antibiotics to treat the fish disease, but we know that the antibiotic resistance is a critical issue. So they want to use some biocontrol approach instead of the antibiotic treatments. But the challenge is the phage is not very stable in the aquaculture environment due to the relatively higher temperature and also the humid environment. So in this, and also the phage is difficult to get into the gastric digestion systems in the fish body. So in this case, we are developing some biopolymer formulation to stabilize the phage, then help the phage to get into the intestinal system of the fish body. So this work can enhance the stability of the phage during storage, and it can control the release of the phage in the salt water, and also improve the bioaccessibility of the phage, and also reduce the use of antibiotics. And this work was highlighted by the trade journal. Also, after this work, we are also developing helps the BASF to develop some biopolymer formulation because they want to use the biocontrol approach for the treatment of the plant disease. So on the farm, there's also 2 challenges for the phage formulation. The first one is the desiccation. The second one is the sunlight, because sunlight is will kill the phage very quickly. So in this case, we also developed the very low-cost biopolymer formulation, combined the whey protein isolates and food-grade nanoparticles to prevent the phage against UV and desiccation. So this formulation can work very well for 4 hours exposure to sunlight. And in this case, on the farm, we can just spray this formulation once per day, and then this formulation can be effective for more than 4 hours. So another work is related to the computational modeling of the food processing equipment. So my PhD study was focusing on the numerical modeling for the to optimize the design of the processing equipment, because sometimes we know that, for example, the pulsed electric field treatment system, their treatment chamber are very small, and it is very difficult to measure the parameters in the chamber. So we use the numerical simulation approach to understand what is going on during in the chamber during the treatment, and to improve the treatment uniformity and also reduce the loss of food quality. So the recent work, we are collaborating with Taylor Farms and Terati, because they are very huge produce industry, and as we said, they just usually they just add more and more chlorine into their washing tank, but they don't know how the chlorine is distributing in the tank, and how fast the chlorine will be depleted. So in this case, we developed the numerical modeling and to help them understand how chlorine was depleted in their washing flume. So regarding the future work, I would still be interested in develop some integrated bio-based and low-cost approaches for the food processing, and also for the food contact materials, and also for the using some nature-inspired discovery and design to improve the safety, quality, and sustainability. And also, I think the collaboration is very important, and especially with the industry partners. So I would be more than happy to collaborate if you are interested. So that's all for my today's seminar, and thank you. I would be happy to take any questions you have.
Well, thank you, Kang. That's very good. I see that nobody's put some questions in the chat room at this point, but I think it's fantastic. Well, I have a question I can kickstart. When you were doing the liners, how did you said you use the liners to reduce the loadings on some of the fresh produce? How did you put the product on the liners? I was interested in that.
So, yeah, so the liner work. So usually they will use this kind of plastic containers for the fresh produce, but usually the industry doesn't have the time to wash these containers. So we can just put the liner plastic films between the at the bottom of the containers. And then if we get one new batch of the fresh produce, we can change one. And also these materials are rechargeable. So we can just put into a chlorine solution, and then maybe after 1 hour, we can take it out, and it can be repeatedly used.
Okay. Any other questions from people on the call? See, I haven't seen any come through. I mean, he covered, okay. So a question from Dean is, have you done any sensory evaluation post-treatment?
We have not done that. But for the fresh produce, quality, we test some texture, the color changes, and the phenolic compounds, and also the chlorate, perchlorate content after the treatment. But we didn't do the sensory evaluation. Yeah.
Okay. Any other questions from anyone? So you're continuing to do that work with the, is it with US, one of the big food companies around the phage work, that's work's continuing at the moment?
Oh, this project has ended, yeah.
Oh, it has ended, okay.
Yeah.
I've got another question. So with the food contact service applications, are these likely to be rechargeable as well?
For the food contact surface application, are
Yeah.
those likely to be recharged as well? Yes. So for example, I would say all my work, if we immobilize the chlorine compounds onto the surface, then those surfaces are all can be recharged. So if we use other compounds like the quaternary ammonia salts, then this will not be recharged.
Okay.
Yeah. For example, this plastic surface, it can be recharged. And also, if we immobilize the particles on the plastic surface, this surface can be recharged. But in the first part, I talked about the stainless steel surface, which has the anti-fouling and antimicrobial material properties. This surface cannot be recharged. Yeah.
Okay, great. A question from Graham is, with the strawberries work, did they have to have complete direct contact with the film, like shrink wrapped? Or how?
So if we want, because this work is based on the chlorine-based compounds. So if we want to kill the bacteria on the strawberry surface, then it should be direct contact. But usually we just want to prevent the cross-contamination. So in this case, the test is not based on direct contact.
Okay.
Yeah. So usually, because for the strawberry, it's not possible to cover every strawberry using plastic material. Yeah. So it's not practical.
No, I understand. Dave wants to know if you've done any work with Campylobacter.
Sorry, can you repeat?
Dave wants to know if you've done any work with Campylobacter.
Campylobacter?
Campylobacter, yes.
In the US, I didn't work with the Campylobacter, but currently in my lab, we just started a project related to Campylobacter because I know the Campylobacter is an issue for New Zealand. So we just started one project related to Campylobacter.
Oh, okay, great. And also Barbara wanted to know if you've done any work on Hep A and berries.
We didn't work with the berries, yeah.
Okay.
But yeah.
Okay, and the another question from Haley is the plastic recyclable after use. When you do use that plastic, I think she's referring to.
This kind. So for the plastic materials, we tested the rechargeability for more than 15 cycles. So at least this material can be recharged for 15 cycles. But after 15 cycles, we have not tested that.
Okay. And a long question from Harry is that he appreciates your seminar, great seminar, and particularly the use of that produce approach. He wants to know if you keep track of the non-produce service modification work of the likes of the Fraunhofer Institute and McMaster University, to avoid the use of chemicals, natural or otherwise? And what are your thoughts on this?
Sorry, let me open this question.
Okay, so can you see the question? So basically he wants to know if you keep track of the work of the Fraunhofer Institute and McMaster University to avoid using chemicals.
Okay, so I think in my study, we are using the natural food grade compounds, which is just derived from the food grade, which is already, some compounds are already existing in the food. So we are targeting using this kind of compounds, yeah, not using the external or additional chemicals.
Okay.
So, yeah.
So one other question here is from Catherine at Zesbury. Have you done any work, she wants to know if you've done any work with curcumin to prevent storage disease?
Storage disease.
Storage, you know, sort of diseases that happen during storage.
Yes, we have done some work related to the shelf life, yeah, or the storage. Yeah.
Yes. I thought that was really interesting that you've done some work with curcumin being sprayed on in the supermarket or under the.
Yeah, because this approach is very practical. Even you are, I would say, even at home, you can add some curcumin into some of your foods and then just turn on the light, then it will kill the bacteria. Yeah.
Okay, yeah, no, that's great. Thank you for that. And one other question from Dean is, are these technologies in commercial use yet? I know you've got a patent for some of them.
For the first part, the particle sanitizers, there are 2 companies currently are testing our product in their processing facilities, but still not commercialized. Yeah.
Okay, and another question, how resistant are the stainless steel surfaces to conventional alkaline cleaning detergents regarding their antimicrobial activity?
This is a good question. So for the stainless steel surface, we published a paper related to the stability of this coating approach against the conventional alkaline and acid-based sanitizers, different sanitizers. So the results show us the alkaline cleaning detergents would be better than the acid-based sanitizer. So our approach can work very well if you use the alkaline-based sanitizer. Okay. So in that
paper, could you send me the reference to that paper and I'll put it on the website, our website?
Oh, sure, yeah.
That would be great.
So yeah, in that case, we tested the four cleaning detergents from Ecolab, and I can send you that paper. Yeah.
That would be great. Thank you. Okay, I think we're sort of out of time. So thank you very much, Kang, for a really interesting seminar. I've had lots of messages coming in saying they found it really interesting. And really appreciate everyone who called in today. Our next seminar is on the 29th of May at 10 a.m. So all the seminars are held on the last Friday of the month. So do mark your calendars. And I look forward to seeing you all then. In the meantime, as our lockdown continues, kia kaha, kia maya, kia manawa nui. Be strong, have courage, and keep safe. Thank you.
Yeah, thank you.